Good morning, good afternoon, or good night to everyone, those who are listening to my video right now. My name is Kirandeep Kaur, and today I am going to give an explanation on a biological topic, lac operon. So, first of all, what is lac operon? So, lac operon is a set of genes that are specific for uptake and metabolism of lactose and is found in E. coli and other bacteria. In lac operon, a polycistronic structural gene is regulated by a common promoter and regulatory genes. It is an inducible operon. Why it is an inducible operon? Because it normally remains off. It operates in the case of prokaryotes, and this works for the lactose consumption. And it was explained by Jacob, Monod, and Wolff. And as you can see the picture of three of them down there. Okay, so let us study about how lac operon functions. So there are two scenarios. So now we are going to study about the first scenario first of all. What happens in the absence of lactose? However, lac operon functions in the absence of lactose. So here our lactose is inducer. Okay. So first of all, let me explain you the structure. The structure which you can see here in the video. This is the structure of our lac operon. So first of all, here it is written our P. So what is P here? Our P is promoter gene. So here our P is our promoter gene, and promoter gene is the site where our RNA polymerase is going to attach. And this is our RNA polymerase which is attached with the P, which is our promoter gene. And this DNA will get transcribed. Our RNA polymerase is going to help in transcription. So our next one here is our I. So what is I here? Okay. So here our I is what is I? So here our I is inhibitor G. So here our I is our inhibitor gene, and this is going to synthesize an accessory protein. So as you can see here, this is the accessory protein which is going to be synthesized by Ig. And let's move on to the next, which is also our P, or you can say a promoter gene. Another is our O. So what is O here? O is our operator gene. So here O is our operator gene, and operator gene is the one which is actually going to regulate the expression of the structure genes. So now you all might be wondering that what are structure genes? So let me tell you that in our lac operon there are three structure genes, and these are our Z, Y, and A. So these are our three structural genes which are present in our lac operon. And here our Z gene is going to transcribe and translate in beta galactosidase. So it will produce this enzyme. And our Y gene is going to help for the synthesis of another enzyme, which is known as permease. Okay, permease. And our permease is an enzyme which increases the permeability of the cell to beta galactosides. And the last is our A gene. And our A gene is going to transcribe and translate for the synthesis of another enzyme, which is called transacetylase. Transacetylase. So this is our transacetylase. So let us imagine the scenario in the absence of lactose. What will happen if there is no lactose? So as I have drawn here the membrane. This blue boundary is our membrane, and here our lactose is not present. So here in this scenario, our lactose is not present. So if lactose is not present, then we don't need structural genes, as these genes are used to break down the lactose. Okay, so it is very logical to say that if we don't have lactose, then what's the need of these genes, these three structural genes? Because these three structural genes are generally going to be used for the breakdown of lactose now. And because the transcription and translation is an energy requiring process, and why should a cell waste its energy when there is no lactose to be broken down? So the structural genes should not get transcribed. Well, our switching on and off of the gene is done by our inhibitor gene, but the regulation is hence controlled by our lactose anyway. So here is our I gene. Here you can see I gene, which is controlling the switching on and off of the lac operon. So this I gene here undergoes transcription, and after undergoing transcription, it produces an mRNA, which is known as repressor mRNA. And this mRNA undergoes translation, and a protein is going to be synthesized. And the protein is known as our repressor protein. Or you can also call it as a inhibitor protein. Okay, inhibitor protein. So well, it's your wish what you're going to call it, inhibitor protein or repressor protein. Okay, the protein which is synthesized here binds with the operator. Now this protein goes and binds with the operator, as you can see in this image, that it binds with the operator gene, and due to this our RNA polymerase does not get space to slide over it, and because of this the structural genes will not get transcribed. Okay, so this is the scenario I have explained to you. Okay, what happens in the absence of lactose has been explained to you so far. So now let's come to the next scenario. Okay, what happens in the case when there is lactose? Okay, when lactose is present in the growth medium of the bacteria, what will happen then? So here you can write here, this is our lactose and it is present in this scenario. So what will happen? This lactose will diffuse inside the cell. This is our lactose, okay, and this is our allolactose. I'll explain you this later, okay, why I had written here allolactose and lactose separately. Okay, I'll explain you in few minutes, just wait. Okay, So this diffusion, the diffusion of what? The diffusion of lactose is helped by the permease enzyme. Okay, the enzyme which is synthesized by our YG. But now you all might be wondering that how it is possible. Before the transcription translation, how our permease is present already in the cell. How this can be possible? So let me explain you how this is possible. It is possible because at any given time, the cell has some molecules of permease which allows the entry of lactose. So there is no questioning about it and no worrying about it that how this is possible. This is possible. Okay, so now you got it. So after diffusion, this lactose get converted into another form of lactose, which is known as allolactose, which I had written here. This allolactose is another form of lactose. Okay, and as the inhibitor protein. So here, this is our inhibitor protein, or you can also call it as a repressor protein. Our inhibitor protein has a very high affinity for what? For allolactose. So this is our allolactose. So here our allolactose is also known as inducer. So as I told you earlier that our lactose is inducer. I told you now our lactose is inducer, and now you might all be confused that initially I told you that our lactose is inducer and now I'm telling you that our allolactose is inducer. So let me explain this. Our allolactose is another form of lactose. So it is also referred to as inducer here because lactose is inducer and it is another form of lactose. So it is also an inducer and it is also referred to as inducer here. So we will call it inducer here. So as we know that our inhibitor protein has a very high affinity for the allolactose, so it attaches to it instead of the operator gene. As you had seen in the previous case, what had happened? Our repressor protein attaches with the operator gene, but now in this case, as our inhibitor protein has a very high affinity for the inducer, or you can say for the our inducer, or you can say for our allolactose. So now it get attached with the allolactose, and now our operator gene is free. And as our operator gene is free, so here as I told you here our RNA polymerase is present. Okay, RNA polymerase. So here as our operator gene is free and now our RNA polymerase is also free, and now it can slide over the structural genes, and here our transcription will takes place, and transcription will result in the formation of mRNA, and this mRNA is known as lac mRNA, and then translation will takes place, and as a result of translation our beta galactosidase, permease and transacetylase are formed, and these three enzymes are the enzymes which are actually going to be used in the breakdown of lactose into glucose and galactose. Okay, so now let us take a scenario here. Okay, let us take an example here. Okay, so let's imagine there are 100 molecules of lactose present in the growth medium outside the bacteria, and when they get diffused, when they get in the molecule, one lactose molecule get converted into allolactose and the rest will remain as it is, means the rest will remain as lactose. Okay, well, so these 99 lactose molecules will be broken down into glucose and galactose by these three enzymes, okay, which are present here. But now what is remaining? Our remaining is our allolactose. Now this is left here. And as we know, our allolactose is also a form of lactose. And if it is also a form of lactose, then this is also going to be synthesized by the same three enzymes now. And this is also going to be converted into our glucose and galactose by the function of these three enzymes. And as soon as the digestion of allolactose takes place, now our repressor protein is free again, which who had high affinity for allolactose, and as soon as it is free, it will go and binds with the operator gene again. It will go and bind with the operator gene again. And as soon as it binds with the operator gene, again our RNA polymerase gets blocked. Again this function is blocked, and this transcription process stops. And now no further enzyme will synthesize. So this is how it functions in the presence and absence of lactose. I had explained you the whole process. Hope you understood it well. So thank you so much. Have a nice day.